we would see the sarcolemma, the plasma membrane of the skeletal muscle cell invaginating like this. And these are the T-tubules. As you can see, here we have one myofibril, right? And in the myofibril, we have thin and thick filaments. So when we look at this, we can see that the T-tubules, which were named T because they go deep into the cell in a transverse, T for transverse way, we can see that each T-tubule has this blue structure on each side. Can you see that? These blue structures are containers that store calcium ions. And these containers receive the name of terminal cistern. And within each terminal cistern, we find calcium ions. Now, one T-tubule with a terminal cistern in one side and another terminal cistern on the other side adds to three structures together. And that's the reason why one T-tubule and two terminal cisterns form what we call a triad. Like a tricycle has 3 wheels, a triad has 3 structures. It has one T-tubule and 2 terminal cisterns. And now we are finally ready to go over how a motor neuron triggers skeletal muscle cell contraction. Skeletal muscles are the muscles that we have conscious control over them. And we consequently decide if we are going to contract or not on a specific skeletal muscle. And what arrives in the skeletal muscle cell is the terminal button, the end of an axon that belongs to a motor neuron, correct? Because motor neurons are the neurons that cause motility. And you learned in the nervous tissue lecture that all motor neurons have a multipolar shape. And you learned in the spinal cord and spinal nerves lecture that somatic motor neurons, which are the neurons that innervate muscles that we can consciously control, have their neuronal cell body in the ventral gray horn of the spinal cord. And then we have the axon leaving the central nervous system and going all the way to the periphery. And at the end of the axon, we will have the terminal buttons that will synapse with the specific skeletal muscle cell. So this axon right here is the pathway through which an action potential, a nerve impulse, an electrical signal will flow until this stimulus reaches the terminal buttons of the axon. And the terminal buttons are filled with vesicles that have neurotransmitters inside. Now, let's zoom in right here. So here we have the sarcolemma, the plasma membrane of the skeletal muscle cell. Right here we have the invagination of the sarcolemma, which is what we call T-tubule. And we would see right next to the T-tubule the terminal cisterns, which are filled up with calcium ions, right? This is the terminal button of a somatic motor neuron. And within the terminal button, we have vesicles filled with neurotransmitters. And as you learned in the nervous tissue lecture, when there is an stimulus, this stimulus is transduced, is transformed into an electrical signal, because our body communicates via electrical signals and chemical signals. And this electrical signal is what we call an action potential or a nerve impulse. And that is literally the cell depolarizing. So when the cell is capable of changing from being polarized to being depolarized, from being more negative on the inside compared to the outside, to become more positive on the inside in relationship to the outside. And this change in voltage is the electrical signal. And this electrical signal flows from the dendrites of the neuron, past the neuronal cell body, and then goes away in the axon of the neuron until it reaches the terminal buttons of the neuron. While this electrical signal was flowing through the nerve fiber, this electrical signal, which is a change in voltage, was causing the opening of voltage-gated channels. Those are channels found in the plasma membrane of the neuron that open due to a change in voltage. That's why they are called voltage-gated channels. And when this electrical signal this change in voltage arrives at the terminal button. It causes voltage-gated calcium ion channels to open. Those are calcium channels that open due to a change in voltage, voltage-gated calcium ion channels. And when the voltage-gated calcium ion channels in the terminal button open, they will allow calcium ions that are outside of the cell to flow into the terminal button, to flow inside the cell. And when there is this influx of calcium ions into the terminal button, that triggers the vesicles that have neurotransmitters inside them to fuse with the plasma membrane of the terminal button and release the neurotransmitters into this gap that we have between the neuron and the skeletal muscle cell. And these neurotransmitters are the chemical signals that transfer the information from the neuron to the skeletal muscle cell. Because remember, two excitable cells never touch. Two neurons never touch. A neuron and a muscle cell never touch. There is always a gap. And what will transfer the information from one excitable cell to the other excitable cell are chemical signals, which are neurotransmitters. Now, the neurotransmitters that transfer the signal from a neuron to a skeletal muscle cell is called acetylcholine. And the short for acetylcholine is ACH. So, acetylcholine released by the neuron flows through this gap, which is called synaptic gap or synaptic cleft. And these acetylcholine molecules bind acetylcholine receptors found in the skeletal muscle cell plasma membrane, which is called sarcolemma. Now, these acetylcholine receptors are channels. And they are channels that they open when a chemical ligand, like a neurotransmitter, binds them. Since these channels only open when there is a chemical ligand binding them, they are called ligand-gated channels. And when the acetylcholine chemical ligand, the acetylcholine neurotransmitter molecules bind the acetylcholine receptors, which are the ligand-gated channels, the ligand-gated channels open. And when these ligand-gated channels open, sodium ions that were outside of the skeletal muscle cell go inside of the skeletal muscle cell through these ligand-gated channels. Consequently, these ligand-gated channels are allowing the influx of sodium ions. That's why these ligand-gated channels are called ligand-gated sodium ion channels. Because when they are open, due to the ligand binding the receptor, they allow sodium ions to pass through them. Now, the influx of sodium ions into the skeletal muscle cell is an influx of positive ions into the skeletal muscle cell. And that causes the inside of the cell to become more positive than the outside of the cell. It causes the depolarization of the skeletal muscle cell. And this change in voltage is an electrical signal. It is an action potential. So this influx of sodium ions triggers an action potential in the skeletal muscle cell. And this action potential flows through the sarcolemma. It flows through the plasma membrane of the skeletal muscle cell, including the part of the plasma membrane that goes deep into the skeletal muscle cell, which is what we call T-tubule. Now, the T-tubule, together with two terminal cisterns, one in each side, form a triad. And what happens is that as this change in voltage goes down into the T-tubules, goes deep down into the skeletal muscle cell, this change in voltage causes the opening of voltage-gated calcium ion channels that are present in the membrane of the terminal cisterns, which are containers that store calcium ions. And when these voltage-gated calcium ion channels open, calcium ions that were stored within the terminal cisterns are released into the cytoplasm of the skeletal muscle cell. Calcium ions are released into the sarcoplasm. And when calcium ions are released into the sarcoplasm, into the cytoplasm of the skeletal muscle cell, these calcium ions bind troponin, which changes conformation. Consequently, it moves tropomyosin out of the myosin binding sites of the actin protein. And now that the myosin binding sites of the actin protein are exposed, myosin can bind it. And when myosin binds the actin, it uses one ATP, and then it pulls the actin thin filament towards the center of the sarcomere. And then using another ATP, myosin detaches from the actin thin filament. And this is how our skeletal muscles contract. And with this, we finish the muscle tissue lecture. Please let me know if you have any questions, and I will see you in class. Bye!